invitation. It's nice to be here, and I'm glad I picked the condor to talk about. But you know, my favorite is Indricotherium. Does anybody know that? It's an extinct rhinoceratid, so in the rhinoceros family. And it was 12 feet tall at the shoulder, and its neck could extend up to 30 feet. So it browsed the tops of trees. There were hundreds of there's hundreds of species of rhinos that were extinct, and we're down to just 5 or 6 species of rhinos today. But we'll get onto that in a moment. This is an important topic. It's a deep topic. But with a title like Conservation, De-Extinction, and the Future of Life, you know, I couldn't also just help think about the Hitchhiker's Guide to the Galaxy or something like that. How can any of us talk about the future of life? But on the other hand, how can any of us not? I mean, oughtn't it be a subject that engages us? And it's not an easy one to engage in. Everybody's interested in what's for dinner, you know, am I going to get my taxes done? You know, evolution hasn't crafted us as a species to think long into the future, and yet we are now involved in activities that really demand that we take a much longer perspective. So, with apologies already, you know, I'm going to say what some would expect me to say. You know, is all we have to do is put this stuff in the freezer and we've got it done. And but that's not my viewpoint, actually. I want to broaden the perspective. I think it's very important to put things in the freezer, but I want to explain to you, I hope, a little bit about some reasons why that might be important. And the real method of conserving biological diversity is to save ecosystems. And it looks a lot different from the previous slide. It looks a lot different from, you know, many programs in the way that we're involved now. But you know, if we were good at saving ecosystems, we wouldn't have all of these species going extinct. We're having some problems meeting these goals. And it's fortunate now that the global community is making goals to address how to making standards and or expectations, Aichi goals, and how to address this problem on a global basis. Our country is one of the great minority in the world that isn't participating in these discussions because we didn't sign the Convention on Biological Diversity. But we have a major role to play and are going to be involved in those discussions in one way or another. So, you know, Peter mentioned the grizzly bear and the passenger pigeon, and you know, most of these species within my lifetime have been gone, have disappeared. So, as a conservationist, we have this backdrop of sense of loss, and this has really been discussed strategically as debilitating for the conservation movement. Young people that say, I've grown up hearing it's too late, or you know what can be done, and you know, that's really an issue that has to be addressed. So that's why discussions like this are really important, and it's really a pleasure to be invited to speak in a forum of the humanities. I really appreciate that opportunity, and I expect you're going to ask me some really tough questions. But just to digress for a moment, you know, we have, there's one of these species up here, this one right up here at the top there, that was a bird that was only discovered in 1978 by a student field course in Hawaii. They found a bird that wasn't in the guidebook and told their professor, and who had declined to hike up to the altitudes he asked them to go. So they brought some experts back, and they discovered a new species, the po'ouli. I'm a member of this Hawaiian honey creeper family. Well, it's extinct now. The last individual died around Thanksgiving in 2004. And the discussion about what do we do with this population, how many are there, do we leave them there, what do we do, should we bring some into captivity, let's count them again, the numbers kept going down. It's a real tragic story. I'm sure Peter knows it well, but it's a real example of dysfunction in our addressing issues. But for reasons that don't need going, I don't want to, we were given a task or the opportunity of receiving the last individual of the species after it had died. And we were able to grow cells from it. So these are living cells of a species that's gone extinct. And we didn't have these cells in the laboratory and growing until after we knew we collected the sample and the last one was gone. So it was a surreal experience to come into the lab and say, how are these cells going? And we take them out and look at them, and they're dividing and they're doing well. And then we can harvest, we can prepare chromosomes. We're the first people to see the chromosomes of the species, and how bittersweet to think that, it's something that's gone. So I would dearly like to see these issues addressed in a more positive way, because the frozen zoo isn't a mausoleum. The frozen zoo is a resource for trying to do studies that prevent extinction, that answer some of the questions that stand between our ignorance about the biology of the species and knowledge-based action on their behalf. So I want to talk to you about the California condor. It sounds like a lot of you know a great deal about it, and I know the local zoo has been very active with condor. The Natural History Museum has a repository here in town of condor materials, and you can see California condors in the Santa Barbara Zoo. But just to quickly go over, one of the things was, like the Po'auli I told you about. I was in graduate school, and there's a big debate about how many condors are left. And some people say, I think the number's this, and we've counted this, but we think there are probably more. And this went on for a long time. And it wasn't until the early 1980s that there was actually, somebody said, well, let's count them. Let's not guess, let's do a better job. Let's put people out there and count them. And so they had to try to find marks that would identify individuals, like, patterns in their feathers, who's molting, that kind of stuff. And they came up with these estimates, and the results were shocking, that the numbers were so low. And then ultimately, 3/4 or the breeding pairs, one member of every breeding pair disappeared over one winter, or 3/4 of the breeding pairs. And so it was clear, only when it was clear to everybody that they were facing extinction, was a decision made that will bring them into captivity. And it was a controversial decision. There were lawsuits and so forth. But it saved the species. But we got engaged, which of these are male condors and which of them are female condors. We developed tests to do that because they're externally monomorphic. So that's put us in a really kind of an interesting position to be involved in the condor project right from the beginning, and to have a sample from every individual. So we have a refrigerator in my lab, and it has DNA from every California condor, including the ones that, the chicks that hatched and then subsequently died. So it's over 700 samples. So one of the questions was, who's related to who? Because you want to try to avoid breeding close relatives. We didn't know how old these birds were. Their plumage becomes adult when they're 3 or 4 years old, and after that you can't tell if they're 3 or 4 or 30 or 40. And how could we, the hope was to have offspring from them. So we did an approach using DNA analysis. It was called DNA fingerprinting that was available in the 1980s, and it identified that all of the condors were basically like 3 clans, we call them. Larger families, some of them only first-order relatives, that is, parents and their offspring. Well, more recently, we've been able to correct that. We've been able to correct that or amend it or refine it because the genomes of California, of 36 California condors have been sequenced. We've described all of the variation of a species in terms of DNA polymorphisms. That's almost entirely true. If there's any genome biologists here, you can ask me and I'll explain the difference. But so that's now created a circumstance that has really changed. So suddenly we're in a different era, and it's emerged rather rapidly, although some of us thought it was coming and would be coming, but now it's upon us. And so there's these new insights that are coming from being able to study whole genomes. What we learned about our own history is a demonstration of what you can get out of these kinds of studies. And I would feel that that we want to do this for other species besides ourselves, because the benefits in terms of health management or understanding evolutionary potential or learning more about the biology and history of a species are readily apparent from the studies of the species that's been studied the most this way, which is ourselves, our species. So we're learning things right now that I think are really exciting from the genome aspect, and it's showing that the population growth back in time, the way the numbers fluctuated, whether there were repetitive bottlenecks or there was a long period of time with a small number of animals, have a profound influence on what the trajectory or what the genetic risks are for a species today. And there are methods that look like that we can look predictively at that, and we can look at that and assess a species to make an understanding about whether, for some species, low number, they get along at low numbers. Other species, they continue to decline. We may have a more predictive science in this field, and that's only one of the applications. But another thing about condors is that they have a genetic disease. They have a disease that causes abnormalities of development. So chicks like this, the legs and wings fail to develop, can't hatch out of the eggs, and it's a lethal dwarfism kind of disease. And because we have the genomes and because we designed the study in the way we did, we now have developed a carrier test to identify who carries this deleterious gene. We know the gene has to be carried by parents of chicks that are affected. But we know that there must be other individuals in the population that are carriers that haven't had the fact that they possess the gene shown because they produced an affected chick. So developed a way to do that, much in the way at the dawn of the human genome project. Cystic fibrosis was mapped and mutations were identified, and so that risk assessments could be given to couples who had a sibling or a relative that had cystic fibrosis, and they would know whether they were at risk or not. So this is a pedigree of California condors, and down at the bottom here is this big long sibshit, this large family, and the red ones are ones that died of chondrodystrophy. We know that, for those of you that, if you're from your genetics, Mendel's peas, you know that recessive genes, you breed two heterozygotes and you get 1/4 of the offspring that are affected. You get 1/4 of the offspring that have 2 copies of the wild type gene, and you expect half the individuals to be carriers. We didn't know who the carriers were, but now we're identifying it. We find out now that some of the individuals who were founders were also carriers. And so we're now tracing which of these males have had offspring, and to trace the carriers down through the population. We want to know whether the gene frequency of this gene is higher in the wild population or the captive population, or whether some of the pairs that are reproducing in the wild or that didn't reproduce in the wild one year, were they at risk of producing a chondrodystrophic chick. But the real issue for condors isn't this genetic disease, it's the environment. They're not growing. The rate of population growth is being sustained by releases from the captive population, because lead in the environment is affecting California condors. 65% of all condor deaths are tied to lead mortality. Governor Brown signed AB 711 last year so that we'll eliminate lead bullets in the state of California. It'll take until 2019 to implement it. But we're the only state that's doing that, and California condors range in Arizona and now into Utah, and they're also being introduced into Baja California. They're also threatened by diseases. They're susceptible to West Nile virus. So every California condor is captured at some point in its life, even if it's a chick that fledged in the wild. There's multiple reasons for that. One is they want to know what sex it is. So they take a sample and we help them to identify what sex it is. And the other is they want to give it a West Nile virus vaccine so it won't be susceptible to West Nile virus. There's another threat to the condors, and that involves wind energy development with all of the large wind generation turbines that are going in. The areas that condors prefer to use are areas that have high wind flux, high wind generating power potential. So there are conflicts at hand. But I don't have time to get into it, but there are ways that we're hoping that those can be addressed. But some of you are saying, like, how did you get that picture? Because there is a condor that doesn't have tags. That's one of the ones that hatched in Baja. And we never could my colleagues, never could capture it. Took a long time to capture it. But they were dying to get a picture of a condor without. They liked the pictures of condors without the tags as much as anybody else. But the tags, in some ways, save their lives because when we see that they're behaving abnormally, we can catch them up and test them for lead. And at any one time, every year, about 20% of the condors have had episodes of acute lead toxicity that require they be inpatients and given chelation therapy. So the idea of this free condor flying in the wild is a bit mitigated or altered by the fact that they're all regularly caught up to assess their lead levels. They have to be immunized against West Nile virus, and they are provisioned to try to reduce their lead loads, but that's not wholly successful. But here's a project where now a species that was on the brink of extinction, we are working to recover it, and we have a new vision of its biology through a study of its gene pool, not as an abstract entity, but as detailed DNA sequence information. I think we're going to see more of that as genome sequencing becomes more prevalent. But for these populations that are small and are showing problems, we would like to, and there are questions about their gene pool or signs of inbreeding. There are circumstances where they are considered, where they have been doomed and lost, or there are other circumstances where intervention has occurred. And one of them is called genetic rescue, and the Florida panther is an example where this regional name for what we call a mountain lion here, or a cougar, or a puma, was showing signs of inbreeding depression, and animals from a different subspecies, but the most closely related one, were brought in, females were brought in and bred with the native Florida panthers, males. They produced several litters of cubs, and those females were then removed, but their cubs revitalized the population, caused a spurt of population growth. So genetic rescue is a real phenomenon. We know that sometimes species that are small in numbers can be rejuvenated or rescued. And this has been done by moving animals, and it's happened with wolves, it's happened with where they were natural migrants, went heroic distances, and then all of a sudden the population that was dwindling and declining grew rapidly. It's been done with bighorn sheep. It's been done with a variety of species. The possibility would exist to do this with other methods. It's been done with black-footed ferrets by artificial insemination, where early animals from generations past had their semen saved, and now they can breed with individuals many generations down the pedigree and restore genetic variation. This could also use some of the kinds of techniques that have been developed in human reproductive science and used in animal science, including in vitro fertilization and somatic cell nuclear transfer. And it could theoretically be done using stem cell technology. So maybe by now some people are getting uncomfortable about the technology moving in. We'll stay tuned. So I want to talk a little bit about the black-footed ferret. It was thought to have gone extinct in the only known population at the time in Melite County, South Dakota, and after 1974 none were seen. But then in 1985, black-footed ferrets were found again in Wyoming. And there was in 1985, there was a wild population that was found, and some of those individuals were captured and brought into captivity, which was a controversy about that again. There was a big discussion, but only a portion of them bred. But the wild population all disappeared, died from canine distemper. So from a subset of those animals that were present in Wyoming, and a subset that were caught and that survived, because some of the animals that were caught died immediately of distemper. The sampling was taking place even as this infection was wiping them out in the wild. So now they've been bred in a federally funded facility, the Black-Footed Ferret Conservation Center outside of Boulder, Colorado, and they produced over 8,000 young. There are 300 in the wild now, at the most, and there's been real problems in keeping the ferret populations going in the wild, and I'll mention that in a minute. But what's happened is that all ferrets today now come from 7 individuals, and they're related basically as half-siblings, and they're not going to get less related. But wait a minute. Artificial insemination from a ferret that was collected that was alive, that was an offspring of one of the wild populations, turns out to be, if it was semen from a living male, would be genetically valuable because he's now not so closely related to all the individuals and restores genetic variation. But there's another way too. In 1985, we received skin biopsies from black-footed ferrets in Wyoming. One of them subsequently died from distemper, left no offspring. The other produced offspring, but it died without leaving offspring. And then she died without leaving any more offspring. So there's no living ferrets that are descended, that we can say are descended from these 2 animals that are present as cells in the frozen zoo. So we've asked the question recently, what benefit could come, from if these cells were actually, if there were living animals instead of these cells. So before proposing that we do something, let's just measure this. So we sequenced the genomes of those 2 cell cultures and sequenced the genomes of the 2 current-day ferrets, and we can show that we can show what is predicted, but we can show in greater detail because we can say much more about the genetic variation that's been lost. Genetic variation has been lost. We can say how much, we can say in what kinds of genes, and we can make more quantitative estimates. So this is a good lesson, although maybe a severe one for those of you that aren't familiar with talking about single nucleotide polymorphisms and genome analysis. But there's a domestic ferret whose genome has been sequenced. So when we sequenced the black-footed ferrets, we could align the sequences with domestic ferrets. And so then we knew something that gave us a great deal more information. We found millions of variable sites that were different, sites that were different in the black-footed ferret than in the domestic ferret. But when we looked to see how much of the variation that was lost, most of the differences between the ferrets are in the portion of the genome we don't know what it does. And that's a case study in our ignorance. And there are these, it's Cheerio and is the ferret from the current generation, and these two on the bottom are the ferrets on the wild generation. So you can see that their numbers are bigger down here. But you might have hoped maybe intuitively that there was going to be some big signal about survival and so forth. There's no signal there. It's waiting for the next generation of genome bioinformaticians to parse these data out. And we're going to have to learn a lot more before we're going to say, oh, this causes them not to do well. But the options for using these cells. So here's the chronology, and here's the amount of genetic diversity. And we start off in the beginning, and we've got the 7 founders, and they're losing genetic diversity. They've lost 14%. Now, captive breeding programs to preserve gene pools are generally the benchmark, as we say, we want to save at least 90% of the diversity. So that's already been missed. So that's been a concern. But what can be done about it? It's only going to get worse with only 7 founders. With the crossing of using the artificial insemination from the early generations, you can increase that. You can increase the amount of genetic diversity that's retained, but you still reach a lower level. If we were able to have those cell lines produce a living animal, they would have a much higher amount of genetic diversity. And maybe this would be a genetic rescue for the black-footed ferrets. The black-footed ferrets are alive now. They're not extinct, but they have, there's no genetic disease they have, but there's questions about their robustness, about their fitness. So could using the genetic diversity from the animals in these cell lines help rescue them? How would you do that? It would be by cloning. That's the only way to do it. But domestic ferrets are cloned regularly. Domestic ferrets are used as a model for pulmonary pathology associated with cystic fibrosis and pulmonary edema, which is often an end-stage problem causing death from influenza. So there are domestic ferrets that are used to study these human medical problems. And there have even been transgenic domestic ferrets produced, where the gene causing cystic fibrosis in humans has been put into a domestic ferret. And in domestic ferret cells, those cells cloned so that living animals, living domestic ferrets, have the human form of the gene. And those can be used for studying drug efficacy and various types of treatment for patients with cystic fibrosis. But the fact is there are laboratories, at least one I know of, where they can take a ferret cell and make an animal out of it. And they can tell you what it costs. Somewhere about $50,000 to $100,000. It depends on how you figure it and how much overhead you get charged and the kinds of things that the administration understand. But, that's not de-extinction. That's genetic rescue. But it would require these same kinds of methods that people were talking about for de-extinction. And so we think it's really important to be saving these samples. We think that the samples we have in the frozen zoo, which are entirely vertebrates, they are mostly mammals, but includes a lot of reptiles, and we're very proud to say we're really increasing the number of amphibians because people haven't been working on that, and amphibian conservation is a huge issue. But this is an irreplaceable connection collection. We could not recreate it. Most of the animals in it are dead. One of them is extinct, the Pohoruli. One of the things that I like to tell people about, because it's a really unsung accomplishment, is, if you look at the repository of DNA sequence information, it's called GenBank. Every GenBank accession has a page that talks about the source, who put the information in there, publication credits, or who was involved in the work, where it was done, what the source of the material was. If you query that GenBank resource for San Diego Zoo, you find over 500,000 hits, and it's going up. The frozen zoo has provided an extraordinary role in expanding our understanding of the natural world. And it's contributed to hundreds of conservation-related studies, and now stands to be able to contribute to genetic rescue, or at least we're talking about that. And I want to say, I've talked to you about some species, but in extreme cases, for some species, the only way that genetic rescue might be accomplished is going to be from these cells, because other methods haven't worked. That suggests to me a couple of things. One is we ought to be saving more cells, because there's species that are in trouble, and if nobody saves the cells, we can't talk about this method. Of course, we want to be trying to not have to use these methods, but I can only tell you that the opportunity to use these things becomes more common on a regular basis. And I want to talk to you about the end of the spectrum, which is the northern white rhinoceros. There's only 5 of these animals left. They are 5 living. The southern white rhino almost went extinct at the turn of the 19th to the beginning of the 20th century, but it was rescued, and technology for expanding the populations basically by translocating them, because they had reached carrying capacity in one place, and they figured out how to, it's not easy, it was a big task to figure out how to move rhinos. They don't walk into crates, and you can't lasso them. So it took chemical immobilization technology to advance the methods to do that. It was done by a South African who passed away recently, Ian Plater. The northern white rhinoceros, was never counted. We don't know whether it was once more numerous. We do know that there were a lot of them. We know that a century ago, our president, Theodore Roosevelt, traveled with some of his family and went to Africa, and they shot some of them, and you can see them in the Smithsonian. And now the species became a focal point of concern. A national park was set up for its last habitat, but this is in the area of South Sudan, Democratic Republic of the Congo. Civil strife there is wildlife. There was just a story recently about this park about all of the elephants getting poached in a short amount of time. It's absolutely heartbreaking. But the northern white rhino is already gone. The 7 that are left are, well, I said 7, you see, because it's only been a year ago there were 7, now there are 5. And when I made this slide, the living ones were in color, and now I've had to strike out 2 of them in color, Angelique through there and Sydney here. But these are the ones that are left. But we have cell cultures from 12. We have enough of a gene pool, I want to argue, that we could preserve the species. None of the species is doomed on its own because the females are aged, there's no reproduction. They've been trying to reproduce them. There's no reproduction going on. So it's an example of the need to expand our notion of what extinction is about, because there are 5 animals walking around, but the probability that they're going to be around for any length of time is nil. So can something be done? Well, with colleagues at the Scripps Research Institute, we took these cells that are in the frozen zoo, and they were turned into induced pluripotent stem cells. So these are cells that can make any cells in the body. And in the mouse, it's been shown that you can take cells that were started the way we did and turned into induced pluripotent stem cells basically the way we did. And you can further direct them by managing their tissue culture environment. And they can produce eggs and sperm. So we could theoretically rescue this rhino with this kind of astounding technology. So we've mapped out an action plan. You know, this sounds like, oh, we could do this, we could do this, we could do this. We don't know if we could do it, but we think we're going to try. We think we should try. And here's where good discussion takes place. Should we try to do this? One of the questions comes up, well, gee, shouldn't it be better to spend the money on something else? I don't think the money to do this would go anywhere else. I think the money for this would come from people who want to see if we can do this kind of genetic rescue. But it's very important we have discussions about this. And we are in an era where we are learning on a very regular basis how our ability to manipulate genomes and our tools for understanding their makeup and their evolution are getting more and more sophisticated. Yet, biodiversity is increasing, is decreasing. We have discussions about making, I've talked to you about, maybe you didn't know that there's transgenic ferrets. Maybe you don't know about everything that's going on in the world of commercial agriculture, where the selection in dairy and beef cattle is going, and I'm not even talking about plants, which has which I guess you're going to talk about some, but this is bioengineers of the future may have a different perspective about what the range of possibilities might be. We could, I can speculate, but maybe we'll do some of that at a later point. But I think we're we're going to be challenged to understand the magnitude of what we might accomplish, and it's going to be crucial that we consider how we want to direct our efforts. And this is going to involve, not glib statements. It's going to have to involve really careful thoughts about what kind of world we want. So I think it's really important that young scientists, conservationists, poets, everybody get involved in a discussion about this. This is a northern white rhino, Nayan, was born in 1989. I happened to be there the week she was born. I took this picture. I didn't realize at the time how much it has influenced me. But here is this precious baby rhino. At that point, maybe there were 30 or 40 or 50, maybe there were more of these animals left on the planet. Now she's the last female to reproduce. In 2000, she had a baby. This precious baby rhino with her enormous mother, these are big. They're this tall at the shoulder. They're huge. Enormously powerful animals with a sense of grace that you might not expect a rhino to have. They get dissed because they don't see well, but they are acoustic specialists, olfactory specialists, and they are ballerinas. They have 3 toes that they run on. And they can run at 30 miles an hour and turn in a body length. That's as good as a cheetah. Cheetahs run faster, but they can't turn any quicker. That this group, the tenderness with which they all met impressed me deeply. And we don't, biologists, don't talk about tenderness. So, forgive me. We haven't really talked about saving those kinds of processes, and the only way to do that is if one of these, if they need more of their kind. So that's my goal, is to have a tenderness time, But now let's move on in the last minutes, because the elephant in the room is not an elephant, it's a mammoth. You know, maybe that's what you came here to talk about, and I've sidelined you with ferrets and condors and tenderness. No apologies. But there are a couple of things about this. It excites people, you know, and the possibility to delve into the past and learn about the lives of these species by sequencing their genomes, and then to think that we could edit a current day species, the cells of a current day species. You need cells. When people say, oh, we can do the DNA, that's a shorthand, incomplete thing. You need, you know, you're never going to de-extinct anything except by doing it in a cell. Cells are life. It's got to happen in a cell. So it'll take place in a cell, and that cell will have to be made into an animal. And whether you can make the same animal or not, my work with that I just showed you here with the ferret says, you know, like we don't know where to start. I mean, okay, mammoths have a different hemoglobin molecule than Asian elephants, their closest relatives. But if you made an Asian elephant with mammoth hemoglobin, you would have an Asian elephant with mammoth hemoglobin. In my book, you wouldn't have a mammoth. So what are the millions of changes between mammoths and Asian elephants are going to be the important ones? Well, you know, of those millions of changes, most of them are going to be in parts of the genome we don't know what they do. So I'm not sure how to do that. But you know, the other part of this issue is, you know, it's the attention that's focused around this. It's hard for a scientist, you know, to watch these movies and love them and see how your kids love them and get so angry that they have this animal that eats acorns that has the completely wrong dentition. It's just an assault on your intelligence. But this is the world that children are growing up in. This is the world that we're going to have to say, you know, is going to be the deciders about whether we say, listen, why don't we just engineer an Asian elephant? You know, or why don't we just engineer a cow to be kind of, you know, with a longer neck, and we'll call it a giraffe. The tools to do that are at hand. The decision about the appropriateness of that will be influenced by the marketplace. It'll be influenced by, but it fundamentally will be influenced by human guidance or of what kind of world we think is acceptable. When the mammoth was sequenced in 2009, an article came out in New Scientist saying, well, geez, what could we sequence and then bring back? And this list frightens me. It's first place, it's just crazy. I mean, it really, you know, like talking about bringing back Neanderthals is a real ethics hot button, to put it mildly. But then to talk about bringing back a giant ground sloth, I mean, okay, they were very interesting animals. Who are you going to gestate it in? There's just this idea about suitable surrogate, or, you know, I think, you know, we thought that the DNA might be the problem. The methods of getting the DNA are getting much, much better. The ancient DNA sequences, there was recently a paper published looking at the sequence of a 700,000-year-old horse. So the DNA may be less of a problem, but you know, we don't know how to do this reproduction. Very, very few cases, and I don't know that would we want to. So I'm sure you'll be talking more about that. I've had the joy of talking about the work of many collaborators, my colleagues in San Diego, people at the Smithsonian, who worked on the condor, the people who actually did the genome sequencing at Penn State, and the groups that funded this. You know, there's no way you can get money from NIH or NSF. This is weird stuff, and it's outside. But there are individuals and foundations and zoo supporters that let us approach this work, and in a way that we hope will reflect the interests of people in the future. Thank you.